Great. All right. All right. Well, I think we're live. So thank you everyone for joining our second in our CAN-ARX webinar series. CAN-ARX is a new project that SEDS-Canada is debuting, and it's focused on bringing Canadian university students opportunities to conduct meaningful and real research in Canada's Arctic. So we have a really special guest here today, Paul Sokoloff. We have him from the Museum of the Nature Museum of Canada, and he's a senior research assistant in the Botany and a member of the Arctic Flora of Canada and Alaska project. And Paul's work boils down to cataloging plant and lichen biodiversity in the Arctic and beyond. Beyond being a little bit more relevant here, given the space theme. So any given day, he may be in a faraway place, such as the Arctic, doing field work in the museum's herbarium, studying specimens, or taking part in outreach activities on behalf of the museum. He first came to the Canadian Museum of Nature as a master's student, and he helped determine that the Fairmelds, not sure if I'm saying that right, Milkovich is not a plant species all on its own. And 2 days after submitting that thesis, he was off on a plane bound for Victoria Island in Canada's Western Arctic. Since then, he's been on many Arctic field expeditions with the museum, and as well participated in the Mars Desert Research Station as crew there. So today, Paul will talk about some of these Arctic and Mars-related research and experiences. So with that, Paul, take it away.
Wonderful. Thank you so much, Kevin. It's really great to be here. SEDS and CAN-ARX is doing amazing work, and it's an honor to be invited to present on behalf of all this. As Kevin mentioned, I'm a botanist. I'm not really a space scientist, so this is kind of fun for me to stretch a little bit. If you're a fan of Star Trek: The Next Generation at all, I'm more Keiko O'Brien than the rest of all of that. So kind of that's the lens to look at this presentation through. So I'm going to start sharing my screen here, and there we go. You should see the title slide with a paraphrasing Mark Watney, because it's pretty much impossible to talk about space and botany together without discussing the Martian. And I'm not that kind of botanist. I don't grow things for a living, and I'll tell you a little bit more about the kind of botany that I do later on. But first thing I'd like to say is that all science is done as a team sport, right? All science that's worth doing in any case. And so this is not just the work that I'm doing. This is the work that our whole team is doing, including folks from the Mars Society, so Shannon Rupert and David Murray, biologist Sam Macbeth, outreach specialists like Mike Irvin, colleagues at the museum like Jeff Sorella, Colin Freebury, Paul Hamilton, and Troy McMullen, and project members from the Mars Arctic 160 mission that I'll discuss a little bit later, Anushri Srivastava and Yusuke Murakami. And the other thing I'd like to say is that today I'm coming to you from Ottawa, which is the traditional unceded territory of the Algonquin Anishinaabe people. I'm very grateful for the ability to live and work on this land. And the research that I'm showing to you comes from both the Arctic, so Inuit Nunangat in the Canadian Arctic, and from the area around the Mars Desert Research Station, which was Ute and Paiute land. So without further ado, I'll come into what a botanist does. So most people, when they think about botany, they think about, well, especially space folks now, right? We think about agriculture, and that's agronomy. It's a branch of botany, but that's not what I do. Lots of folks think flower arranging. That's a florist. Very different job, very rewarding, I'm told, but not quite what I'm into. No, I'm a systematic botanist. So what that means is that I'm interested in the classification of living organisms. I like sorting things for a living. So I work on essentially classifying plant and lichen life based on various different characters. So morphological, so how the structures of a particular plant, do they go together or not? Genetic evidence, genetics tells us a lot about the evolutionary history of organisms, so we can use that to sort plants into categories. And all of this work happens at the Canadian Museum of Nature. Now, if you've ever been to Ottawa, you've probably been by our building downtown, which is this large castle that you see here. And I actually don't work in this particular building unless I'm doing an outreach event. Most of my work actually happens at a research and collections facility over in Gatineau, Quebec, which houses about 14.6 million natural history specimens that we can use to essentially say what organisms could be found where at any particular place. So those are the materials with which we use to classify life, and that's what I do on a day-to-day basis. Is I work with those specimens, conducting scientific research on what plants grow where in the Arctic. And that's all, that's a lot of lab-based research, that's a lot of herbarium-based research, but the part that's nearest and dearest to my heart is field-based research. By working with this amazing team here, we're seeking to categorize Arctic plant and lichen life, and then tell the world about it through scientific publications and also through public outreach presentations, which is the nice thing about working in a museum, is that science communication is built into our job. All right, so how do we essentially work to classify that plant life? Well, I mentioned that we use morphology, we use genetics. It all comes from those collections, those 14.6 million natural history specimen collections I mentioned earlier. And to do those, we actually have to go out and collect them. And so on any given summer, you can find members of our museum Arctic Flora research team essentially scouring the tundra in and apart different areas of Nunavut, Northwest Territories, in and across Arctic Canada, and essentially from base camps, we hike out and collect at least one example specimen of every plant species that we see, and increasingly every lichen species that we see as well. As you can tell here, there are predators about. It is polar bear country, so we are typically armed, or we have polar bear monitors from local communities come out with us just to make sure that we're safe while conducting our research on the land. And then once we've done a full day of collecting, we'll bring those specimens back to our base camp, and we'll squish them in between cardboard and sheets of paper and put them out in a plant press just like this. This is state-of-the-art technology circa like the 1700s. It hasn't changed much in the last couple hundred years, because pressed plants, once they're flattened and dried completely, they retain a lot of the morphology that we can use to examine them, to examine what species they are, and they last for a very, very long time. At the museum, our oldest collections are from the 1700s, but the oldest collections known to science actually come from some of the tombs in ancient Egypt, so thousands of years old. As long as they're preserved well, we can use them in perpetuity. And we have, out of those 14.6 million natural history specimens, I keep forgetting the million, we have about 1 million pressed and dried plants in the herbarium. And this is a library of specimens that we can go and use to essentially figure out the answer to that question: what grows where across the Canadian Arctic? And by keeping everything forever, we can build off of the work of previous generations. We can go back and say, oh, this species was actually known here, was not known here, or at least not collected from that area at a specific time. It also means that all sorts of folks can use the herbarium for all sorts of research. People use it to track flowering times of plants, because as long as the label with each pressed flattened plant specimen includes the location and the date, we know something about how that species occurs in place and time. And by putting specimens in the herbarium, we can always go back and check our work later, or update the taxonomy of specimens, their names, as our understanding of their evolutionary relationships evolve. So at the end of this talk, I hope that one of the things that you will take away is that if you're doing any sort of biological science on Earth, and I think this is true of exoplanet stuff as well, or within the solar system, et cetera, put your samples somewhere safe where people can use them for generations to come. And as we're thinking about, sample retrieval missions from the current Mars 2020 rover, we're going to think about sample curation pretty intensely. And this is something that we do at the museum every day. All right, so that's a little bit about what I do as a botanist. And now I want to tell you a little bit about what our work in the Arctic involves. And so most folks, haven't had the opportunity. If you're in Southern Canada, you probably haven't had the opportunity to go to any part of the Canadian Arctic. But we talk about it a lot. So you probably have a pretty good idea in your head of what the Arctic is, or at least a picture of it. And it might look something like this. You might think, ice sheets, icebergs, maybe a zodiac full of tourists on a trip. This is all true. This is a picture I did take in the Canadian Arctic and Baffin Bay during an expedition with students on ice. But that's not all the Arctic is. The Arctic is actually quite a large area in Canada. Everything above the tree line, which a botanist like myself would consider Arctic, or at least the habitat of Arctic species, is about 40% of Canada's landmass, over 2 million square kilometers. So in all of that area, it's easy to imagine that there are so many different kinds of landforms, different geological histories, different biological histories. So different habitats and different landscapes which host different species of plants and lichens to search for if you're interested in that kind of thing. But firstly and foremostly, the Arctic is homeland. For hundreds of years, a lot of how science has looked at the Arctic is as this frontier and as this vast terra nullius. It's never been like that. It's always been Inuit Nunangat, the Inuit homeland in Canada. And by working with local communities, we can significantly enrich science. We also therefore have responsibilities to form reciprocal relationships with our communities when we're working there. And so, it's really important to me that we're doing research right when we're up there. So these are the examples of, these are the 4 Inuit regions in Inuit Nunangat. So there's Nunatsiavut in northern Labrador, Nunavik in northern Quebec, the territory of Nunavut, and the Inuvialuit settlement region in the Western Canadian Arctic and Northwest Territories. And by working together with communities, we do better science for everyone. So by doing a lot of this field-based research, this lab-based research, and by going back to the collections that we're increasingly digitizing and going through, in a systematic manner, we think there are about 800 different plant species in the Canadian Arctic. And I say we think because there's not a definitive number. Every time we go up, we add new species to the known flora of the region, just by examining places that haven't necessarily been inventoried for biodiversity before. And we do this about once every year, or once every 2 years. We'll mount an expedition to collect plants to say something about the botany of that area. And we'll typically target under collected areas in the Canadian Arctic by just essentially plotting up all of the collections we have on a map and looking for places that are underexplored, or working in partnership with communities that are inventorying regions that are interesting to them. Like this last summer, we went to Agutini Territorial Park on northern Baffin Island. So now that I've told you a little bit about the Arctic, I'll go back there in a second. You might be interested to know what does an Arctic botanist like myself have to do with space exploration? And a lot of that actually stems from something that happened way back in 2013, which really seems these days like a lifetime ago. Back in 2013, the Mars Society, which is an organization based out of the States that advocates for the crewed exploration of Mars, put out a call for people who are interested in potentially participating in simulated Mars expeditions to their habitat in the Mars Desert Research Station in Southeast Utah. And with the idea that this would be a test run for a longer mission to their facility on Devon Island, which you might know, the Flashlight Mars Arctic Research Station at Houghton Crater, which is next to the Houghton Mars project that's run by the Mars Institute. So when I saw this, I thought that little part of my brain that was always like loved space, but took this turn into botany, I was like, well, I could maybe throw my name in the hat and see what happens. So I did, and a few months later, I got this email. Oh, hey, we've selected you. Do you want to come join a 2-week mission to Mars, the Mars Desert Research Station, to test your suitability, to work with a crew, to see if you can do a long-term mission, but also to conduct science while you're there. And I said, yes, absolutely. And so in fall 2014, I actually joined MDR's Crew 143, which in addition to me, was comprised of these fine folks here. And we were actually a multinational crew with folks from Russia, the States, Canada, and France. And none of us had actually ever met each other before we arrived in Utah, which was amazing because we all hit it off really well. A lot of us still talk to this day, which is incredible. And during our time at MDRS, we were essentially living and working as if we were on the surface of Mars. And so lots of pancake breakfasts. I imagine it, when we do some crude exploration on Mars, we'll probably have at least 1 or 2 pancake breakfasts. We had a few more than that, but we were in sim, right? So what is it your standard day in a Mars simulation at MDRS look like? Well, most mornings and nights consists of lots of reporting. So writing reports for mission control, doing some cooking, cleaning, just kind of standard life stuff that will come up when we do any sort of crude exploration of space. Except, with folks that you've never met before, or at least in my case, most crews actually know each other before they arrive at MDRS. But that's not what most people see. Most people see the simulated space aspect to what happens at MDRS. And so, for a lot of crews, the research that they do, it can be more human factors. Okay, how do we live and work in this space and maximize efficiency, or how do we train people on different things? Medical crews will often go to try new techniques or to test new equipment. Engineering crews will often go to test new technologies. And in fact, there's a University Rover Challenge that happens at MDRS every year. With the pandemic, it hasn't been on for a few years, but fingers crossed it'll come back soon. But if you're a field biologist or any sort of field scientist, you're typically going to spend most of your time trying to get out on extravehicular activities outside of the hab. And our crew was actually pretty heavy into geologists and biologists. And so we did lots of simulated walks on the surface out and around the habitat. And so, whenever we'd go out, we'd put on a simulated spacesuit. We'd have to go through, as you can tell, it's a bit of an ordeal. This is Anastasia. I was kind of like, we worked together, making sure that we had a complete mission plan, and we were working under similar constraints as to what we think it would be like when we're doing it on Mars itself. We go through the airlock. You have to do the simulated decompression cycle. Please, as you can see in the image on the far right, please don't forget to close the airlock when you come back in. One of the best movies to watch while you're here is Total Recall, by the way. Like, I highly recommend. But that's neither here nor there. But when we're outside of the station, then we're working in these thick glove suits, trying to do whatever tasks we have been assigned. So, we did lots of repair tasks in and around the station while we were there. But when we were heading out on science, we got onto our rovers, which are ATVs, heading out in different areas, doing whatever scientific research that we'd been assigned. And in my case, that was botany. Now, I didn't do botany in like lots of crews do, which is where they work on growing plants in simulated Martian regolith. The crew before us actually had a radish experiment growing, which is this picture I had taken on our first day at the hab. No, the botany that I proposed and was accepted by MDRS Mission Control was actually a botanical inventory of the area around the station. Because while it definitely looks Mars-like, it's got those red rocks, it's very deserty, it's also a very rich botanical habitat. It's got all of these different areas that harbor really interestingly well-adapted plant life for the desert. And it just so happens that the director of MDRS, who's since become a really good friend of mine, is a field ecologist by training, and she's interested in biodiversity studies in and around the area. And so she said, yeah, go for it. If you can collect plants and lichens under Mars mission simulation restrictions, do it and then see what you find. And so every time I went out and I collected plants, in my gloves, digging them up carefully, taking notes so that I could make the herbarium labels later, and getting back to the hab. Instead of using the lab at the station to look at rocks, I was actually identifying the plant specimens that we brought in and pressed them just like we did when we were working in the Canadian Arctic. And you can inventory pretty much anything like this, including cacti. You can press the cacti that grow in and around Mars Desert Research Station. And so you just have to kind of fillet it like a fish first and scoop out the insides and press it. And don't use your glove. Even if you're wearing spacesuit gloves, you'll get full of needles forever. And you'd think you wouldn't have to tell a botanist to be careful, but it's true. So that's what we did. We did a biodiversity survey of the plants around MDRS during Crew 143. But since then, it's actually grown into a multi-year Mars Bio program where multiple crews have gone to do biodiversity research in and around MDRS, both in simulation, like the first time, and then outside of simulation, just using the Mars Desert Research Station as essentially an Earthbound Research cabin. And this is interesting because it kind of expands the mission profile of MDRS. It allows us to do science there that is analog Mars science, but not necessarily under simulation conditions. So we can, essentially do space stuff there increasingly. What we do is definitely not analog space science. It's definitely Earthbound biodiversity survey stuff. But what that does is that allows us to build comprehensive databases of the flora that grow in and around MDRS. And this allows us to inform all sorts of analog astrobiology missions and analog space missions that'll happen at the station moving forward. By better understanding the station as an Earthbound habitat, we're better informing Mars analog missions that happen later on. And I mentioned that, it's very Mars-like, but it also has these interesting habitats. The desert, when we talk about astrobiology on Mars, we talk about follow the water, right? This is, if we follow the water, we'll find where things can grow. This is true in the desert as well. If you follow the water, you'll find life, and you'll find lush oases of plant life if you find habitats that have a lot more water input. And in exploring all of these unique microhabitats, both in simulation and outside of simulation, we've actually been working on building together a complete list, a hyperlocal flora list of all the plant species that grow in and around the Mars Desert Research Station. So through the years, we've counted about 81 taxa of vascular plants. And so those are things that have roots and shoots, cacti, trees, yuccas, grasses. And I say taxa instead of species because in some cases we haven't identified things down to the species level because we haven't collected it for reasons that I'll tell you about shortly. And in some cases things are subspecies, etc. But that includes, really common desert things like the prickly beavertail cactus, Opuntia polyacantha, and greasewood, which you see here on the far right, which is a really common desert plant you see in riverine kind of water area, water-rich habitats. And there's also things like big stem rabbit rush, Ericaria nauseosa, Artemisia filifolia, which is a sagebrush, very, very common. But then you get small things as well. Members of the lily family, including desert onion, Allium macropetalum, and the white vein lily on the far right here. And I'm sorry if I keep throwing out scientific names. This is just kind of what I do. If you need clarification on anything, I'm very happy to answer questions about plants afterwards. I mentioned yuccas. Yuccas are a really interesting part of that flora because I just think they're neat. But also, we don't really get those in many places. They're sporadically found in and around the station, and so they were exciting finds whenever we did find them. Those are all perennial species that grow in and around MDRS, but there are annual species that grow in and around the Mars Desert Research Station as well. And in fact, if you've ever seen pictures of the super bloom that can sometimes happen in the deserts of Southeast Utah, these are the species that you're looking at, Phacelia demissa and Cleomella palmerina. And they're just beautiful purples and yellows. This is a hotspot for diversity of milkvetches, members of the Astragalus genus. So these are 3 species that you found here. Kevin, in my introduction, you mentioned Fernald's milkvetch. It's actually in the same genus as all of these. So finding these growing in and around MDRS was a bit like coming home for me, finding plants that I knew and loved well. And then I mentioned that we don't always necessarily know what an individual species are because we don't necessarily make it collections. Sometimes we actually will document a plant growing in an area just with a photo. And in the case of this particular cactus here, a sclerocactus, a fishhook cactus, we didn't collect it because all of the fishhook cacti around MDRS are actually federally listed endangered species, and they're protected by law. And so we don't collect those when we see them, but we can document them and then give the data with those occurrences to the Bureau of Land Management so that they can better protect these species. And on the far right, actually, this is an interesting plant. This is Hoffmannseggia repens. It's a member of the bean family, and it is actually only known on planet Earth from the San Rafael swell area, from in and around the Mars Desert Research Station area. So it's interesting because we use it as a place to learn about living on Mars, but it's also a habitat for plants found nowhere else on the planet, nowhere else anywhere. So protecting that area will help us, when we talk about becoming multiplanetary, I sincerely believe that means protecting Earth to the best of our ability as well. And so by better understanding endangered species on Earth, we're better able to protect them. And in that vein, we're also documenting the incidence of invasive and introduced species that grow in and around MDRS. Weedy species are a huge problem in American rangelands and habitats across the American Southwest. And so by better documenting where these invasive species occur through herbarium specimens, we're better able to inform conservation management as well. And so these are some of the invasive species that we documented there, mostly in the mustard family, the 2 on the left, and then an invasive grass on the right. So that's the vascular plant species that we documented while we're there. But we are also looking at lichen species because the lichens are an important part of the flora as well. And this is because lichens are extremely well adapted to grow in extreme environments. They can resist desiccation extremely well. They can resist UV radiation exceptionally well. A lot of this comes from their life history. They're a composite organism consisting of a photobiont, so an algae or a cyanobacterium, and a fungus, and they grow together, in an assemblage that essentially can survive harsh areas. And in fact, the species like Xanthorrhoea elegans, the elegant sunburst lichen, can even survive being exposed as part of a European Space Agency experiment on the outside of the International Space Station. And then it was brought back in and it continued to metabolize. So better understanding these organisms can better help us document biodiversity in the area, but understand biological systems that allow for stress tolerance in extreme environments like you might find off planet Earth. So right now we think there are about, what we've documented, 13 species of lichen in and around MDRS. We know that number is going to be greater. In fact, we're working on a project right now to grow that number. But these are the ones that we've identified to the species level. And in fact, identifying species of lichens is as I'm learning, not as easy as a plant where I can just look at it and say, oh, it's this. You need to run often genetic and chemical analyses in order to come to a determination. And so you can get these big, kind of what we call foliose or fruticose lichens, like Lecanora on the left here, which kind of blend in with the sandstone environment. But one of the most common lichens that we found were the desert fire dot lichen, Caloplaca trachyphylla on the right, that can be found growing in the lee of rocks all across the area around the station. Cobblestone lichens, those in the genus Acrospora, are also very common in and around the area. Interestingly, they actually have spores that are so microscopic they can be carried in the jet stream. And so these species can actually be found in many different desert habitats across the planet, including in the Canadian Arctic. So again, interesting species to study from an extreme adaptation perspective. And I mentioned that we're working on growing the number of species, and a lot of that are coming from the collections made by a nursery, who you can see here collecting during the Mars Desert 80 mission, which happened in the fall of 2016. So she made multiple collections of lichens that we're working on identifying, and that'll be a really useful resource for us to talk about lichenological diversity in and around the station. And Anushree has then since gone on. She's doing a PhD in the UK, and actually she's working on the Perseverance mission. So she's doing some really cool things, and she collects lichens too, which I think is incredible. And then on top of the plants and lichens, we also at the museum have expertise through Paul Hamilton in microbial algae and cyanobacteria. So he's an expert on these organisms. And so we actually were interested in then algae that are endolithic, algae that grow inside rocks in the desert, which are another interesting extremophile to study from a perspective of astrobiology. And by figuring out the taxa that grow in the area, folks who are doing astrobiological analog missions in and around MDRS will get better equipped with just what are they going to find when they get there. And we found about six taxa of endolithic cyanobacteria and algae growing in and around MDRS. All right, so that's essentially our where we're at right now with our analog work at the Mars Desert Research Station. And what I've convinced you of is that by better understanding analog sites here on Earth, especially if we're doing analog missions that involve field biology or field geology, better understanding the biological diversity of those areas can only improve the fidelity and only improve the quality of our analog work. And so, we're kind of a funny group of pretend astronauts. We don't necessarily get really into the simulation because instead of avoiding the plants, we're seeking out everyone we can find. But we're doing this in the hopes that we can better protect these areas and then better inform future analog research to come. And so if you're interested in all of that, especially at MDRS, we've actually published 2 papers so far on that research, and they're open access, so you can go and enjoy, and let me know what you think if you have any questions about that. But a lot of that's all in Southeast Utah. And for what you're going to be doing as part of the CAN-ARX work is all focused in and around the Canadian Arctic. And as you likely are aware, there are some really great analog research sites, and there's some really great analog research happening in the Canadian Arctic, in various parts of the Canadian Arctic, in and around the Canadian Arctic archipelago, I think mostly, although there are some stations at Colony Lake and other places that have missions as well. So I did mention earlier the FMARS, the Flashline Mars Arctic Research Station, which is on Houghton Crater on Devon Island. And that's the one that folks typically think of when they think of analog research, because it is that tuna can on the tundra, to paraphrase Matt Bamsey, who was a previous analog astronaut there. And essentially this place is well known to host visiting Martian missions. And in fact, I mentioned the Mars Arctic 160 missions. I was really thrilled to be invited to be an investigator on these missions, seeking to improve our biodiversity knowledge in and around Houghton Crater, in addition to the various other science objectives that happened during that mission. And so in 2017, our crew at the top left here actually went to FMARS and did a month-long simulation while they were on the ground. Now, they were doing a lot of work in and around simulated spacesuits, just like we do at MDRS. But this time they were actually making sure to do a complete lichen collection, as well as collect some vascular plants as well, to better understand the diversity of that area. Now, I actually haven't been to FMARS. It's on my bucket list. It is a place that I absolutely would love to go one day. But I have been fortunate enough to work in Kauswituk, Resolute, which is the staging point for missions into Devon Island. But for folks who are doing any sort of hierarchic research, this is a place that you will pass through, because this is where the Polar Continental Shelf Project Hub is. And so Coheswituk is essentially a hub of hierarchic research activity in Canada. It's a wonderful community, and if you ever go, I really recommend working closely with the local Hunters and Trappers organization, because they can give you some great tips on where to find things, and just sitting to chat with them with tea is really a lovely experience. And so in 2017, while this crew was on Devon Island, I was actually working with a museum colleague, Dr. Troy McMullen, inventorying the vascular plants of Coheswituk. And now I want to present those results here, because I understand that for CAN-ARX, this is one of the potential project locations, either now or in the future. And certainly if you're working in the high Arctic, these are taxa that are going to be common there, especially in and around this kind of habitat in the central archipelago, which consists of a lot of shattered limestone. So in terms of museum collections at the Canadian Museum of Nature from this area, we have a little over 650, 98 of which I made during my time there. And there are about 75 plant taxa in 13 families. And that's actually lower than the diversity MDRS, which makes sense if you think about, latitudinal gradients. But I'll throw a little curveball here in a minute. Now that's just vascular plant taxa. That doesn't include the lichens that grow in and around the area. And these are just a few examples of the common lichens that grow in and around Resolute. This one on the left, Lechinora marginata, is exceptionally abundant across the rocks in and around Coheswituk. And these lichen taxa are often, they'll grow in cracks on the rocks, but a lot of them will grow directly on the rock. And as my colleague Troy says, they eat the air. They get their nutrition directly from the air, and some from the rock itself. And so we're working to better study these organisms, because they tell us a lot about environmental factors at Resolute. And then in terms of the vascular plant diversity, unlike MDRS, there are actually in Resolute no introduced or invasive plant species yet. Something that we're keeping monitoring for as we go is looking for introduced species in the Canadian Arctic. Grasses are particularly abundant in and around this area. These are all different species of grass, because they actually typically like disturbed habitats, or habitats that are frequently turned over. And the activity in and around the research station has resulted in a bit of disturbance in the landscape, and allow a lot of grasses to kind of find that foothold. But there are a lot of tundra flowers as well. Perias and cerastiums and drabas are these yellow ones that I love, but are taxonomically difficult as all get out. And on the bottom right, a very common site in and around Resolute is the purple mountain saxifrage, which is the territorial flower of Nunavut, and the first flower to come up in the spring. And again, just these are a few more from Coheswituk. There is a very colorful flora in and around the area, even if it is only a few species. But you typically see them in areas that have heavy water and heavy nutrient inputs. And so in places that are just out on the kind of the cobbly scree, you typically find plants growing small and in cracks. Another really important biological component of this ecosystem, and I shouldn't neglect to mention the mosses, which are exceptionally important in Arctic ecosystems, but cyanobacteria, like this nostoc colony that you see here, are really important in Arctic ecosystems, and they're, in fact, they drive a lot of the primary production in these areas. And so we collect these as well whenever we go out and are studying what's on the land. Now, the McGill Arctic Research Station, which we also visited in 2017, is another well-known Mars analog area. Not necessarily because it looks like the planet Mars, but because a lot of astrobiological work has happened at the station and continues to happen based out of the station, looking at microbial life in extreme environments, including gypsiferous springs in and around the station. There's been a lot of plant collecting there because there's been continual research happening there since the 50s. So about 803 collections just from the station, 78 of which we made during the 48 hours that we were able to visit the station on that trip. And there are actually a lot more taxa. There are 133 there. And that's because instead of the calcareous scree that you find in a lot of Resolute, Kaisuituk, and Devon Island, there's actually a lot more cordilleran granitic substrate and heterogeneous, like calcareous and acidic rock types, allowing a lot of different plants to grow. It's also a lot more stable, so a lot more tundra has able to develop over time. Soil development in the Arctic is extremely limited, but in Resolute, it's even more limited than places like the McGill Arctic Research Station. So Arctic poppies like this love to grow. Again, grasses and sedges are a common sight in and around the tundra here, because they like those footholds in disturbed environments, and they can grow pretty quickly to reproduction during the very short Arctic summers. But again, there's a whole lot of other colorful Arctic flowers on the landscape. And you know, a lot of these are either well adapted to attract pollinators quickly, because there are pollinators, there are bees and mosquitoes that pollinate flowers on the tundra, or they're well adapted to self-pollination, because in you know, the short Arctic summers, you really kind of have just a little window to get going and to reproduce. And so a lot of them have varying adapted life histories depending on where you're at. And lastly, before we take some questions, I wanted to mention the vascular flora of Victoria Island, because the Canadian High Arctic Research Station in Cambridge Bay on southern Victoria Island is also a potential CAN-ARX mission site in future, for now or future years. And our team at the museum, led by my colleague, Dr. Jeff Sorella, have actually compiled a complete vascular plant flora of Victoria Island based on collections from all of these red dots that you see here. And that's actually work that we did over several years, compiling 7,000 specimens into a list of 288 taxa that you can bring with you when you're in the field. So if you have any questions about, okay, what is this plant, and you're on southern Victoria Island, this is the definitive resource that will be able to help guide you. And if you're still having problems, that's what your friendly neighborhood space botanist is here for. So if you're doing any sort of botanical work, I'd be very happy to hear about that. Okay, so that was a bit of a whirlwind, but I wanted to make sure that we had time for questions. And I hope that I've convinced you that analog work and traditional biological botany expeditions can go hand in hand, because I think together we learn a lot more about the sites that we are doing space analog work in, if we can better understand them as earthbound habitats as well. With that, I would like to thank you know, not only the museum and the Mars Society, but folks at Nunavut Parks, Parks Canada, the Polar Continental Shelf Program, Polar Knowledge Canada, McGill, the many colleagues at the museum who have made this work happen over the years, the many colleagues at the Mars Society, and of course, all of the communities that have supported our work through many decades of research, and all of you for your attention. Thank you very much.
Well, thank you so much, Paul. That was an incredible presentation. All kinds of, like you said, a little bit of a whirlwind of topics, but it's really cool to see how it all weaves together and it's interrelated. And yeah, you know, it's spaces for everyone. That's what we're here for. So with that, I'll turn it over to any of our project team members. If they have any questions, feel free to jump in and ask. So we have Danya and Sonia here. Do you guys have any thoughts or questions for Paul? It's a similar type.
Can you hear me? I've been trying to make my mic work here. Can you hear me?
Yeah, we got you.
Okay, perfect. Thank you for your presentation. That was really great, and it was just so good hearing about the analog missions. I think that's something that at least all of us, you know, who are interested in the space at some point really like to do it. I guess my question is more about, you know, what the times where you did the analog missions. What did you find kind of the most challenging about those times?
That's an excellent question. I think probably one of the things that I found most challenging, and I think people often find in these contexts, are balancing different research priorities. Because you know, when you're going with a crew, you all kind of have, especially for folks who are interested in this, you're all bringing in projects that you want to get done, and there's typically only so much time in the day or only so many resources you can allocate. So striking a balance to make sure that all of the research happens in the most efficient way possible and effectively for every crew member, I thought was something that we was challenging, but I was really happy to see it worked out pretty well in the end. And a lot of that, I think, had to do with the fact that when we were doing those analog missions, our crew commander, Alexandre Mongeau, was very exceptionally fair in trying to make sure that everyone had the resources they needed to do their jobs.
Thank you.
No problem.
Yeah, Paul, I actually ended up having a question as well. It's a little bit more broad scale, but in terms of actually conducting field work in the Arctic, it's a fairly logistically challenging and technically challenging endeavor. So I was just wondering if you could talk a little bit about some of the stories that you have about actually doing that yourself with all of the experience that you had up there.
Absolutely. Yeah, no, you're right. I did kind of gloss over all of the like how we did it, just to like, this is what we did. But you're right. I mean, in terms of research, that's really important. And so when you're doing Arctic research, it really, especially if you're planning a field season, and for most of us, I imagine that happens during the summer. So in July or August, or maybe even early September. And that's typically when you can get up there. You're actually working the year before to secure logistical support or funding from the Polar Continental Shelf Program or Polar Knowledge Canada, because you know typically the call for support is in October the year before you want to go, and around the same time you're formulating your goals and objectives, and you're also coming up with your plan. That's a good time to do things like apply for permits, and permits are important in the Canadian Arctic, because they're one way that we tell communities about what we're doing, but they're also a way to make sure that if there's feedback that needs to come our way, it does happen. And so you know, depending on where you're going, you might apply for permits to access parks. You might apply for permits from the Nunavut Research Institute or the Nunavut Department of Wildlife, water permits from the Nunavut Water Board, planning permits from the Nunavut Planning Commission, and likely permits from the relevant Inuit association that you're going to be working with. And that can be, you know, a bit of an undertaking. And so if anybody does have like specific questions about permits, if somebody wants to shoot me an email, because you know, I think it's just knowing where to go. But that's also a good time to then actually start building relationships with the communities that you want to work in. And communities really they're interested in work that happens in their own backyards. And they're interested in getting involved in any way they can. And so if you're planning on doing long-term work in an area, building relationships is really helpful. And actually, even before all that, when you're working on research questions, going to communities and saying, hey, we're doing these space things. Are you interested? And what kind of things do you see happening here, can help with a lot of that. So I highly recommend, you know, and I mentioned Hunters and Trappers Associations. Those can be ways into communities, but also Facebook is your friend. And don't just reach out to people saying, oh, hey, you live here, I'm going to come do this. But there's probably a community message board where you can say, oh, hey, you know, we're doing this, you know, and typically, if you're doing research permitting, those officers can help guide you to places that are good venues for knowledge sharing as well. And then when you're, you know, a few weeks out, then you're thinking about, okay, shipping, food, housing, etc. And then I mentioned wildlife monitors, hiring wildlife monitors from communities when you're in polar bear country, which is a lot of the Arctic. So there's a lot that goes into logistical work in the Canadian Arctic. what I will say is that the Polar Continental Shelf Program has a really good guide for a lot of this. Like, what do you have to think about when you're thinking about working in the Canadian Arctic for the first time? And also Interact, which is an EU field station initiative, actually has a complete PDF guide on their website to field planning for Arctic research. And I highly recommend having a read through that.
Wow, that's yeah, that's a great resource. I hadn't discovered that. You said EU, like European Union guide, basically.
Yeah.
Gotcha.
Yeah.
Very
Absolutely.
cool. Wow, yeah, that's some great tips as well. And I really like what you said about working with the community, because after all, it's their backyard, absolutely. And they're not giant communities at the same time. So you will be very close with these people for the time that you're up there. Yeah, we actually are a project team with Danya. They have applied to the Polar Continental Shelf Program. That was part of the reason why we selected Resolute as our top choice for the moment, because it's looks like a great program, and it does seem to be the place to go as a hub for anything seriously Arctic related. So
absolutely. And Resolute is also, it's a great community. So, when you're up there, please, and a lot of this too is to what extent you can do things will be driven by how the state of the pandemic, of course.
Yeah.
And the community's comfort level with everything. But I think the best way to do Arctic research is to do Arctic research in partnership with folks who live there, because they're super interested. Folks want to know what's going on, and they can provide insight that we just don't have.
That's some really great advice, and it's a really great tip, and thank you for sharing that. Yeah, with our selection of Resolute, do you have any fun experiences or any insight into that area that it seems like you have a lot to share on that. So
yeah, well,
anything else on there?
One thing I can tell you is that if you take ATVs across a river, make sure that, if you go in the morning, you come back before afternoon, because as the sun comes up, the snow melt will happen, and you don't want to drown your ATV. And I'm not saying that we almost did that, but I'm just saying be careful when you're out there. And I would also recommend meeting with the HTO there to talk a little bit about that, about what you're interested in. But also, like, the folks at Polar Shelf they're incredible. They do amazing work. And they can help facilitate a lot of this. So working closely with the folks at Polar Shelf is great. What else is specific to Resolute? One thing that is very self-serving for me, but if you're up there and if you're interested in what a flower is, you can actually use your phone and snap a picture for iNaturalist, and then people will actually tell you what it is, and that data will actually go onto GBIF, the Global Biodiversity Information Facility, that can be used for research purposes. So if you're interested in botany at all, you can become botanists while you're there.
That is really cool. Yeah, we part of one of our sub-projects within CAN-ARX is a citizen science activity for the local community up there, where they do a 360 image, and we try and teach them a little bit about some Martian habitation and some of the limitations. It's a really cool program, but the citizen science concept in itself is such a really great way to connect with the community, to get more data points, because that's something that we need as scientists, and really expand the horizon for the scientists doing the back end and the people that get to really experience that as well. So
absolutely
great idea.
Awesome.
Last question on my side. Do you have a favorite Arctic plant?
No, don't make me choose.
Sorry.
Actually, here, I'm going to share my screen again. It's in the presentation, so I can show you. All right, so I can go back a few slides. What's that people say, you can't pick a favorite child, but everyone has one. That's terrible. This one here in the bottom left, the red one, mountain sorrel. The Inuktitut name is Kunguli, and the Latin name is Oxyria digina. And it's my favorite because it's edible. It's full of vitamin C. It's really important to Inuit. And it grows across the Canadian Arctic. It grows in every habitat I've ever surveyed, or at least every
Wow.
location. So that's probably my favorite plant, at least
Multifunction.
today.
That's really great.
Yes,
Yeah,
and
for
it's
sure. Lack of
delicious.
fruits and veggies, it's a real concern in those extreme environments. So very cool.
Awesome.
All right. Well, do we have any last questions from any of the project team members? Danya or Sonia, looks like I see you guys online.
Awesome.
I'm good on my end, but thank you so much again for your presentation and your insight into Arctic research. I'm sure when the time comes, we're going to be using a lot of advice that you gave us here.
Well, feel free to send me an email. I'm really happy to support, and I think this is a really cool initiative.
Sure. Thank you so much again.
Well, yeah, again, Paul, on behalf of our CAN-ARX team, thank you so much for your presentation. It's been really great having you.
Thank you so much for having me, and good luck with the mission.
We'll send you updates.
Excellent. I'm looking forward to it.
Thank you so much.